All right. Well, great. I think we can go ahead and get started. It's right at 2 o'clock. So welcome everyone to our MPRINT webinar for the month of November. We're pleased to have Laura Ramsey joining us today from her new position at Children's Mercy Hospital in Kansas City, Missouri. Dr. Ramsey received her PhD in molecular cellular developmental biology and genetics from the University of Minnesota Twin Cities, and then completed a postdoctoral fellowship in pharmacogenetics at St. Jude's Research Hospital before joining the faculty at Cincinnati Children's Hospital, and eventually rising through ranks to associate professor and co-director of the genetic pharmacology service there. She's recently moved to Children's Mercy, and where she is working as the section chief of Individualized Therapeutics in the Division of Clinical Pharmacology, Toxicology, and Therapeutic Innovation. In her new role at Children's Mercy, she is working towards leading the implementation of pharmacogenomics program that's fully integrated with the electronic health record system. And developing model-informed decision support tools for several medications. And she's also the co-director of their clinical pharmacology T32 fellowship program. Dr. Ramsey is interested in all aspects of pharmacogenetics, from basic research to implementation in patient care, and has several projects that have been funded by extramural awards, including an active R01 from the National Institutes of Health on improving the effectiveness and safety of escitalopram in pediatric populations. And she's also received an Opportunity Pool Award from our MPRINT Hub to work in the area of pharmacogenomics and antidepressants. So with that, I will turn it over to Dr. Ramsey to talk to us about the pharmacogenetic influences on sertraline exposure and tolerability in children and adolescents. Laura, welcome.
Thank you, Dr. Quinney, for the kind introduction and the invitation to present to everyone today. I do have funding from BTG Specialty Pharmaceuticals, but they do not make anything that is related to sertraline or pharmacogenetics. So today I will show you some of our data on how pharmacogenes influence the pharmacokinetics of psychotropic medications like sertraline, and hopefully educate you on how pharmacogenetics can be used in clinical care to adjust medication dosages. So I got this slide from a psychiatrist that I work very closely with, Jeff Strawn, because he always starts his presentations with this to show this spaghetti plot of individual patient data from clinical trials of adolescents treated with one of these 4 SSRIs. So there's some patients that respond very well, and there's some patients that respond almost not at all here towards the top in 12 weeks of treatment. And what he wants to know when he's got a patient sitting in front of him is this a patient that's going to respond very well, or is this a patient that is not going to respond to this medication? This variability can be explained by a lot of things. The demographic factors, the age, sex, weight, clinical factors, the dosing factors, and of course something that you're always worried about with adolescents is adherence and actually taking the medication as prescribed. So there's a lot that goes into the variable medication exposure, but pharmacogenetics is one of the things that do contribute to this, and the variable exposure contributes to the variable responses. So I see a lot of new names on here to me, so I want to make sure that I explain pharmacogenetics well. And in clinical pharmacology, we often use the analogy that the body is a bathtub, and using a standard dose is like turning on the water at the same rate in all patients. And the metabolizer group for an active drug that's metabolized into an inactive drug can be thought of as like the size of a drain. So a poor metabolizer has a very small drain. They are more likely to overflow the bathtub with high concentrations and are at high risk of side effects with standard doses of the medication. As you increase the speed of metabolism, the drain gets bigger, so you're more likely to have no water as an ultra rapid metabolizer or have subtherapeutic concentrations and a risk of non-response. You can also have inhibitors, and that can be like hair in the drain, making someone who's genotypically a normal metabolizer into a phenotypic poor metabolizer, and this we call phenoconversion. There are times when pharmacogenetics is useful, and there are times when there are significant associations with pharmacogenetics, but they're not really going to be useful in determining the dosing or adjusting dosing. And so that's illustrated here with, on the left, a narrow therapeutic range where the pharmacogenetic extremes, the poor and the ultra rapid metabolizers, put you outside of that therapeutic range box. That's a case where pharmacogenetics might help in dosing the medication to keep you keep a patient within that therapeutic range. On the right, there's a larger therapeutic range. There's a significant association with the pharmacogenetics, but it's not going to be very useful to adjust the dosage based on that pharmacogene because all the patients are still within that wide therapeutic range. There are a lot of great pharmacogenetic resources that I want to make sure that everyone knows about. CPIC, for one, provides peer-reviewed, updated, evidence-based guidelines for gene-drug pairs. And they don't tell you when to genotype or whether or not to genotype, but what to do with the genotypes in hopes that one day when we all have our genes known, that these guidelines will facilitate the translation of pharmacogenetic knowledge from the computer to actual patients. And the Dutch Pharmacogenetic Working Group is another group that puts out guidelines, and FDA labels have some pharmacogenetic information in them, ranging from anywhere from don't go over 20 milligrams of citalopram and a CYP2C19 poor metabolizer to exposure might be higher if a pharmacogene for individual pharmacogenes. So it's not, all of them are not very prescriptive, but there are some that are fairly prescriptive in how to adjust the doses based on pharmacogenetics. PharmGKB is a website that takes all of this knowledge and puts it in one place for you. So that's usually my go-to resource in where to find anything pharmacogenetics. And PharmVar defines the pharmacogenetic alleles and the nomenclature. And they have been doing this for several years and have many pharmacogenes represented there. So today I'll focus on Sertraline, or the brand name is Zoloft, because my MPRINT Opportunity Pool Funds award was to study this drug more thoroughly in pediatric patients. So this drug has been around for a long time. There are many dosage forms, so a pretty wide range of doses are used. Excuse me. And it's approved for the use of a lot of things in adults, but the only pediatric indication is highlighted here in yellow, and that's OCD. obsessive compulsive disorder. And so the starting dose and the maximum dose is a pretty wide range of doses here. It's metabolized by multiple CYP enzymes. The major enzymes are CYP2C19, CYP2B6, and CYP3A4, with minor contributions thought to be from CYP2C9 and CYP2D6. I'll show you the pharmacogenetic associations between sertraline and CYP2C19 and CYP2B6. In the FDA label, you can see here that Zoloft or sertraline is a CYP2D6 inhibitor as well. Most of the evidence for pharmacogenetic associations with sertraline has been with CYP2C19, which is a gene that was first characterized in the early 90s and is part of the CYP2C subfamily of genes. There are two fairly common no-function alleles, the star2 allele and the star3 allele. And the star17 is an increased function allele associated with more rapid drug clearance. The star39 is the most recent addition to PharmVar, so we're still discovering new alleles in this gene even 30 years after it was first characterized. So I'll start with some of the studies that have shown the associations in adult patients. There has been two really nice Scandinavian therapeutic drug monitoring studies from this hospital in Norway. So the first one here collected samples from 2007 to 2018. They measured sertraline at steady state and trough and did CYP2C19 genotyping as part of routine care. They also have a laboratory database that had more genotypes, like for CYP2D6 and CYP2C9. And they use this target concentration of 30 to 250 nanomolar. They collected samples from over 1,200 adults and plot here in this graph the harmonized sertraline concentration. So this is basically just a dose normalized concentration, normalized to 100 milligrams a day, which is middle of the road kind of dose. You can see in the graph that the poor metabolizers had much higher concentrations than the normal metabolizers, whereas the rapid and the ultra rapid metabolizers didn't have that much difference from the normal metabolizers. But there was an association with sex and age greater than 65 also. They found that CYP2D6 and CYP2C9 were not significant in predicting these concentrations. Sorry. All right, so the second study was a bit later, 2017 to 2020. And again, they measured the steady state trough concentrations and did CYP2C19 as part of routine care. They had additional targets that they were able to genotype, like this novel CYP2C haplotype that has been associated by their group with reduced exposure to another CYP2C19 substrate, an SSRI escitalopram. They also were able to look at CYP2B6 with the star4, 6, and 9 alleles. And so this study included 840 patients with 1,482 concentration measurements. And so here they are wanting to show the novel CYP2C haplotype, the TG here. And the way they found this variant was to take all of the star1 patients that had escitalopram concentrations that were lower than expected And so they did sequencing and found these variants upstream of the CYP2C19 locus, and see that there is a fairly similar phenotype compared to the well-known star 17 allele. So the patients that have two versions of this TG diplotype, they have the same reduction in exposure compared to the normal metabolizers as the patients that have two star 17 alleles that we consider ultra rapid metabolizers. The patients with one allele, TG allele, though, they have very similar exposure to the star 1s, unlike the patients that have the star 1 and the star 17. They have faster or clearance and reduced exposure, significantly reduced exposure, 17% lower. And then on the other side, you can see, again, the poor metabolizers have over twice the dose normalized concentration than the normal metabolizers. But, you know, you can see a lot of variability here among the star 1s. Still, there are patients that are below the therapeutic range. There are patients above the therapeutic range, and a lot of variability. So they looked at CYP2B6 and found that it does explain some of the residual variability. So now we're plotting the CYP2C19 across the bottom and CYP2B6 across the top. So anybody who had a star 4 allele, they considered an ultra rapid metabolizer. CPIC might break them out into rapid and ultra rapid for those that have one or two, but here they've lumped them together as ultra rapid. And the poor metabolizers are those that are carrying the star 6 or the star 9, two no function alleles, and the intermediates have one no function allele. And so you can see that the patients that are poor metabolizers for CYP2C19 and CYP2B6 have much higher concentrations, and it sort of decreases as you get faster in the CYP2B6. So the CYP2B6 is accounting for some of the residual variability there, and the patients that are ultra rapid for both are pretty close to the bottom of the therapeutic window. Their exposure is 35% less than patients that are normal for both CYP2B6 and CYP2C19. So this is a case where sort of in the middle here, probably, where the therapeutic range is a little bit wider and the poor metabolizers are at or above the top of the therapeutic range. The ultra rapid metabolizers close to the bottom, but maybe still within that therapeutic range. But CPIC did include Sertraline in their recent guideline update for 2023. And their recommendations are for CYP2C19, initiate with the recommended starting dose for normal, rapid, and ultra rapid metabolizers. And for poor and intermediate metabolizers, you can consider a slower titration schedule. For poor metabolizers, a 50% reduction in the standard maintenance dose. And these are both moderate recommendations, moderate strength recommendations. And then when you take into account both CYP2C19 and CYP2B6, it's a little bit like a bingo board where you have to find where your patient is if you have both of these. The recommendations range from, you know, the usual dose or just slower titration, or start low, titrate slowly, and get to 25% or 50% of the usual dose. So they do have recommendations, and that most of these are optional for that includes CYP2B6. In every CPIC guideline, there is a pediatric section now. And in this section that I always look for, it indicates that most of the evidence for associations within all of these gene drug pairs that were reviewed for the guideline, the most evidence exists for citalopram, escitalopram, and sertraline, and CYP2C19. And we can rely on these, or we may be able to translate the adult data into pediatric populations, because the CYP2D6, 2C19, and CYP2B6 activity is equal to adult levels by early childhood. And that's usually when these medications start to be prescribed. But that doesn't always placate the clinicians. They want to see the data in their population before they start using pharmacogenetics in kids. Most of these associations, especially those large Scandinavian populations that I showed you, were all done in European ancestry patients. So how well do they translate it to non-European populations? As Dr. Quinney said in the introduction, I was at Cincinnati Children's, where they have the largest pediatric inpatient psychiatry unit in the United States. They just recently opened this brand new building last month for inpatient care, only for psychiatry, with more than 100 beds dedicated just to psychiatric patients. So they started doing genotyping of CYP2D6 and CYP2C19 as part of routine care upon admission to psychiatry in 2005. So they've been doing that for almost 20 years, and they have been providing these recommendations in the electronic health record. So to date, more than 30,000 tests have been performed. It's all done on site. CYP2D6 and CYP2C19 are the only ones reported when a psychiatry panel test is ordered, and it provides recommendations for 22 medications as a percentage of the standard dose. So before they were reporting anything for sertraline, we did this study on how they were dosing the sertraline, and looked at if there were differences across CYP2C19 phenotypes. So we pulled the data from the EHR. They had access to the CYP2C19 information, but no recommendations for sertraline. And some prescribers never even look at the pharmacogenetic test results. So I don't think they were using them to guide the dosing, even though there was a CPIC guideline that said there is an association. But what we saw was that there was slower titration in the slower metabolizers among these 352 patients with anxiety or depression. There were side effects that were related to dose, but not to CYP2C19. This study and many of the others that I will show you today from our group were done by Ethan Powlite, who was an undergrad when he started with me and doing these studies. So we eventually got around to collecting samples. We wanted to know, okay, well, that's nice that we know the dose is different, but what about the concentrations? So when they get admitted to the inpatient psychiatry unit, they get the genotyping done by either whole blood or a buccal swab, but they also get a CBC often, And there is blood left over from when they do that. And so we were able to pick up the remnant blood samples and measure the sertraline and desmethylsertraline concentrations in these remnant blood samples, and then look in their charts for their dosing history and covariates, and then fit PK model with MW Pharm ++. So in our Cincinnati Children's cohort, we had more than 500 samples from 473 children and adolescents ages 5 to 18 that have been collected from August 2020 through April of this year. And as any psychiatry study, they are more females than males included. And we do have a fairly significant population of transgender patients as well. In Cincinnati, the majority of the population is white, non-Hispanic, and that is what we saw in our cohort as well. We had sertraline doses prescribed anywhere from 12.5 milligrams per day to 250. And we had the sertraline concentrations in the beginning before we got the MPRINT award measured at Indiana University with Dr. Desta. That included the desmethyl metabolite. And then with the MPRINT award, we measured more samples at UCSD with Dr. Momper, and there was no desmethyl sertraline included in that. So our CYP2C19 phenotypes were just about what you would expect given this racial mixture, with 3 or 4% in the extremes and fairly equal numbers of intermediate and rapid metabolizers. There is a significant influence of the genetic ancestry on CYP2C19 metabolizer phenotypes. There are much more poor metabolizers in those people of Asian ancestry that you can see here. There's a very big piece of the pie that is a poor metabolizer here compared to the European ancestry. The lightest piece of the pie here, the poor metabolizers, is very small, but there are quite a lot of ultra rapid metabolizers, and this is very different from the slice of the pie for ultra rapid metabolizers in the Asian population, but among Africans and other populations, there are quite significant numbers of rapid and ultra rapid metabolizers. So for our study, we collected opportunistic samples. We had the dosing time because the patients were inpatient. So all the dosing times were recorded, the blood sampling time was recorded, and we had an alert that fired that let us know that a patient had a CBC result within 24 hours of a dose of sertraline. We're doing this for other medications as well, but they also show very similar distributions here. So most of the patients had a sample around 12 or 24 hours. So those patients, the blood samples are usually taken in the morning for the CBCs. So the patients that take their medication at night, they're in this group. The patients that take their medications in the morning are in this group, or in this one that took had their blood draw at the same time as their medication was given. And so for this part of the study, we found that the doses were similar across the CYP2C19 groups. Whereas our retrospective study before showed that the titration, so the dose at 90 days after initiating sertraline, was lower in the poor metabolizers, we did not observe that in the population where we have the PK samples. So it looked like, even among the poor metabolizers, there are several that are getting 150 milligrams per day, and several ultra rapid metabolizers that are at 50 milligrams per day. We do see that the dose increases with age, just like the FDA label recommended to, initiate at 25 milligrams in the patients that are under 13 and start at 50 in those that are over 13. The clinicians are increasing the dose based on age or body size, perhaps. And it did look like their dose was higher in females than in males, but this was not significant after adjusting for age because, we did have that preponderance of females in this study. There was a much higher concentration dose ratio in those CYP2C19 poor metabolizers, similar to what was observed in the adult studies. I've set up my graphs the opposite direction that they did. So poor metabolizers on the left, ultra rapid metabolizers on the right. And again, more than twice the concentration dose ratio in the poor metabolizers. And it looked like perhaps a bigger influence of the intermediate metabolizers as well, with a 50% increase compared to the normal metabolizers. And 19% decrease in the rapid metabolizers and 35% in the ultra rapid metabolizers. One difference between our study and the Scandinavian study was that our patients were not required to be at steady state. But then we were able to take their dosing history from the electronic health record. It's not always complete, but to be able to account for the time after the last dose and really more closely estimate the trough concentration, the peak, the AUC, and the clearance for each individual patient. And this was done by Ethan on a smaller cohort of 265 samples from 255 patients. And there was no adolescent PK model or pediatric PK model for sertraline that accounted for CYP2C19. So he built one in non-mem, a one compartment model, and found that CYP2C19 was significant on clearance. It provided the most significant reduction in the objective function value, but the rapid and ultra rapid metabolizers were not different from normal metabolizers. Did find that body weight significantly affected clearance and volume, but sex did not. He also reviewed the charts for CYP2C19 inhibitors like oral contraceptives and fluoxetine and found those significantly decreased clearance as well. So when he more precisely estimated the trough concentration, then what we did with just that concentration dose ratio, you can see that those poor metabolizers are very close to the top or above that therapeutic window, with 150 milligrams per day, which is the highest that some of them were prescribed. But there is still quite a lot of variability among the genotypes here. So perhaps we will be able to explain some of that with CYP2B6 later. And one thing to note here is the half-life. He was also able to estimate that. And you can see that it's twice as much for poor metabolizers than for the normal rapid, ultra rapid metabolizers. So those normal metabolizers, they are getting to steady state with 4 doses, which is what the Scandinavian study required for doses. And the intermediate metabolizers, they're going to take more like a week to get to steady state. And the poor metabolizers are more like 2 weeks to get to steady state. And so this is sort of the basis for our recommendation to do a slower titration among those intermediate and poor metabolizers. This illustrates what Ethan found with the influence of weight and the CYP2C19 inhibitors on exposure. So, we found patients as young as 5 up to age 18 were prescribed sertraline. So there is quite a bit of variability in body weight in that age range. And if you prescribe 150 milligrams a day to a patient that's 30 kilograms, they're going to have very different exposure compared to a patient that's 110 kilograms. And that's illustrated here. And then on the right, you can see the difference that having a CYP2C19 inhibitor on board provides. So this illustrates the titration. So using the MW Pharm, Ethan has normalized to a 70 kilogram adolescent with no CYP2C19 inhibitor, what the concentration time curves look like with regular once daily dosing of 25 milligrams and then 50 up to 100, 150, and 200. And again, we had some patients that were prescribed 250 in our cohort as well. So the patients that are poor metabolizers, they are getting within that therapeutic range at 25 milligrams and exceeding the therapeutic range with the 200 milligram dose. And Ethan has also received a F31 from the NIMH to look at whether machine learning can be used to predict side effects and readmissions to the psychiatry unit. And what I'm showing here is his work on predicting side effects. He combined the cohorts that got escitalopram or sertraline. So there were more than 500 patients, and about 1 in 5 has a side effect that we could identify through retrospective chart review. And so what he's showing here is that the AUC, the medication exposure, the clearance, and the time since the last dose increase were the top predictors for whether a patient would have side effects. So lower exposure, slower clearance, or higher clearance, then you're less likely to have a side effect. If you've had a dose change within the last 2 weeks, you are more likely to have a side effect found in your chart. And the AUC for this model was fairly decent for predicting side effects with 0.77. The most common side effect is activation. So this is something that's seen more commonly in children than in adults, where patients are hyperactive and impulsive, have trouble sleeping, have disinhibition. And these patients had 14% slower sertraline clearance than patients that did not have activation recorded in their chart. When we looked at CYP2D6, we found no influence. When we did the PK model, there was no additional reduction in OFE. And when we looked at just the concentration dose ratio, we also saw no influence of CYP2D6. And we have a quite substantial proportion of the population that uses marijuana as well. So there is some evidence out there in vitro mostly that CBD and marijuana inhibit the CYP2D6 and CYP2C19. So we wondered if we would see any influence of this in our population. We looked in the charts to determine if a patient was using marijuana or CBD. There is a urine drug screen that is routinely done upon admission, but there's also the clinicians often ask the patients whether they're using marijuana or not. And so we looked through all the notes for the patients as well. And we found 18% of the samples had been taken from patients that were using or suspected of using marijuana or CBD. And so we had 80 samples that we had sent to Dr. Momper at UCSD for measuring the THC and metabolite. And we found that it was present in 45% of the samples. He also measured patients, looked for these in the plasma of patients not using or suspected of using, and we found it present in only 3% of those patients. So there was, in the PK modeling, we did not have this metabolite and concentration data yet for the THC. So just used the chart review, yes or no, on the PK modeling, and found no influence of the marijuana use in the PK modeling. And when we looked at the metabolite for an influence on the sertraline concentration dose ratio, we also did not see an influence on that, accounting for the other factors like that were significant, like CYP2C19. So we don't yet have the CYP2B6 genotypes, but we they are included on the panel that is used clinically since 2020. And so the clinical lab in Cincinnati is able to pull those from the patients that have had their genotyping done since 2020 among our cohort. It would also be very interesting to look at the 2C TG phenotype that the Scandinavian group found as significant on the sertraline and escitalopram exposure. And then we're also interested in looking at the pharmacodynamic genes, SLC6A4 and HTR2A, on the side effects and outcomes after we adjust for the exposure or the pharmacokinetic genes. These SLC6A4 and HTR2A are often tested for in commercial pharmacogenetic tests, but the CPIC guidelines are that they are not ready for use in clinical care. There may be a class effect with the SLC6A4 and the SSRIs in that the SS patients, those with a short allele of SLC6A4, might have reduced response. But most of the studies that were reviewed, and there were over 500 studies that were reviewed in the CPIC guideline process for SLC6A4 alone, most of them did not account for the dose or the exposure or even have enough power to analyze individual drugs. And so there's no recommendations that are able to be made for SLC6A4 because there's no trials of using SLC6A4 for like drug selection, which could be very helpful in the future if the recommendations might be to avoid SSRIs. This is what a lot of the commercial pharmacogenetic tests say to do. So the associations between the PK, the concentrations, or the clearance, that's pretty easy to find pharmacogenetic associations there with the pharmacokinetic genes. The associations with clinical outcomes there's a lot more that plays into the variability there. Pharmacodynamic genes and environment. There's a lot of stuff that plays into the clinical outcomes. So some have suggested that this means that combinatorial pharmacogenetics is the best for this. But I am here to show you a study in kids that says, no, that's not that helpful. So there was a randomized controlled trial of pharmacogenetics in kids done at the Mayo Clinic in 176 adolescents with MDD. What they showed was there were no differences in outcomes or tolerability at 8 weeks or 6 months between the patients in the genotype group or the treatment as usual group. But what they did see was a difference in the prescribing pattern. So fewer SSRIs were prescribed in those that had the genotype test compared to those that were treated with usual treatment, including fluoxetine, which is the first line treatment for adolescents with MDD. So the combinatorial pharmacogenetics for medication selection really is not effective. And that's what the commercial tests are often used for. So our recommendations for applying pharmacogenetics in practice are to really understand the diagnosis first and whether pharmacotherapy is warranted, what the medication is best suited for the patient in front of you. And then to do pharmacogenetic testing if the medication you've chosen has guidance that's available. So if you're going to choose fluoxetine, there are no pharmacogenetic recommendations. So you don't need to do pharmacogenetic testing at that point. But if you're going to use escitalopram or sertraline, where there are CPIC guidelines, that's when you might consider doing the pharmacogenetic test so that you can use a different dose or titration or use an alternative medication or monitor more frequently. So what I've shown you today is that CYP2C19 significantly influences the sertraline exposure in children and adolescents, but CYP2D6 does not. There are recommendations for using lower doses and slower titration and those slower metabolizers. And the poor metabolizers will likely be over that therapeutic range at that 200 milligrams per day maximum dose that's recommended by the FDA. What we found was that marijuana seemed to have little effect on the sertraline pharmacokinetics. And we are still working on getting those CYP2B6 genotypes to replicate that adult study to see whether the same phenomenon occurs with the additional description of the variability provided by CYP2B6. So there are a lot of people that have contributed to this, most of them at Cincinnati Children's, and are still continuing to collect samples and do the genotyping there. And of course, Jeff Strawn, who I partner with on a lot of studies, and Ethan Powlite, who did most of the experiments and analyses that I showed you today. We have our partners that measure the drug concentrations at Indiana and UCSD. And of course, I want to thank our funding sources like MPRINT and the NICHD and NIMH. So with that, I will be happy to take any questions. Thanks for your attention today.
All right, great. Thank you, Laura. It was a great presentation. And I will ask if anyone has any questions, feel free to put them in the Q&A or chat, or if you raise your hand, we can promote you and allow you to speak and ask your question. So, so I have a kind of a methods question on your PK study that the first one you described that you did looking where you did not see as much of a genetic effect on the dose that they did in the adult patients. Were those patients all at steady state, or were they initiating treatment too, and so maybe had not been dose escalated?
Yeah, it's a range. So there are some patients that are just starting sertraline. There are some that have been on it for a while. There's some that were on it and then stopped, and we don't really know when they last took their medication, other than the dose immediately prior to the sample being collected. So it's not a perfect study by any means for pharmacokinetics, but it is opportunistic and sort of real world, and what we had available to us without sticking the patients again.
Right. And then I know that study you had the metabolite data, correct?
Mhm.
Did you look at the metabolite parent ratios and see any effect there?
Yeah, we definitely saw an influence of CYP2C19 on the parent metabolite ratio. So just as one would expect, similar to adult data and others. There, Dr. Desta was able to measure the CYP2D6 metabolite in some patients. And not as many had detectable levels, but the metabolism for sertraline goes from the parent to the first metabolite, desmethyl sertraline, with CYP2C19 as the predominant enzyme, and then didesmethyl with CYP2D6 as the predominant enzyme. And so we did have that in a few patients, still detectable, but not very many.
Right. So earlier on, you talked about in that beginning of that study, you mentioned that some prescribers never looked at the pharmacogenetic reports. Were you able to convince anyone with your data that the pharmacogenetics might be more important?
Yes, so using Ethan's data from that first study, we did show that to the head of the psychiatry unit, and they agreed that we should add it to the report that is provided. And they did ask that I be more involved in the education of the fellows and the psychiatrists. And so I have been presenting this work at the Child Psychiatry Conference, the American Academy of Child and Adolescent Psychiatry, routinely, to really try to get this into their understanding, and especially sort of contradict a little bit of what the commercial pharmacogenetic testing companies are telling them, that they need combinatorial pharmacogenetics to understand which drug is going to work for them or for their patient. And really working with them on understanding that it's the pharmacokinetic genes that we're at right now is useful. So, you know, you can order just a CYP2C19 test from LabCorp and use that in a going to PharmGKB to see what the recommendations are for the single gene that you're interested in.
Great. Let's see if there's any. I don't see any questions on chat or any panelists or attendees who've raised their hands right now. So thank you. It was a wonderful presentation. I really like the bathtub analogy you used at the beginning, and how you're both looking at genetics and potential drug interactions in developing this phenotype here. So thank you again for joining us today. So
thank you. Thanks everybody for the attention.
Thank you everyone for attending today's webinar, and we look forward to seeing you next time.